The overall goal of these experiments is to rapidly screen and characterize enzymes for antimicrobial activity.
The microslide diffusion and dye-release assay have utility for the functional discovery of new antimicrobials with lytic activity against specific targets, such as a pathogenic bacterium of interest. The main advantages of these techniques are that they are simple to execute, provide flexibility for variation of experimental parameters, and can be performed with laboratory equipment commonly found in most microbiology laboratories.
Prior to starting the microslide diffusion assay, the concentration of the antimicrobial enzyme to be evaluated should be determined using a BCA protein assay kit according to the manufacturer's protocol. Perform a serial dilution of the antimicrobial enzyme using PBS as a reaction buffer. Dispense the protein into individual microfuge tubes and adjust volumes to 20 microliters using PBS. Next, prepare a 0.5% agarose solution by dissolving 0.25 grams of agarose in 50 milliliters of PBS. Heat the solution to a boil until the agarose is completely dissolved. Determine water loss during the melting process by weight and add back to the solution. To inhibit contaminating bacterial growth during incubation of the assay, add 50 microliters of 10% sodium azide in water to the agarose solution and adjust the temperature of the solution to 50 degrees Celsius in a water bath. If viable cells are used as the substrate, omit the sodium azide from the agarose solution. Thaw the heat-killed bacterial substrate on ice. Resuspend the bacterial substrate.